Equilibrium, very important from exam point of view. Reason is 5 mark questions, either they might ask you what is Hardy-Weinberg equilibrium, describe it, or they might ask you mention any 4 factors or 5 factors affecting Hardy-Weinberg. As the concept itself, it's named after the person who put forth the Hardy-Weinberg equilibrium. Probably 2 persons to have put forth this theory, Hardy-Weinberg equilibrium. What you have to remember in a population, I have always told you, in this given class population itself, there is a definite gene pool. What is gene pool? Gene pool is sum total of genes present within a population. All the gene possibilities in a given population or Indian population, as example, the gene possibilities when compared to the Japanese population, the gene pool might be completely different. There might be some changes there. So the sum total of genes that is possible within a given population, we call it as gene pool. Frequency of occurrence of a gene, we call it as gene frequency. What are alleles? A pair of contrasting characters. Like if you take a length of an individual, tall is dominant, dwarf recessive. They are alleles there. For the same character, they have 2 genes. One which is going to have a dominant gene, it exhibits tallness. The recessive gene, it exhibits dwarfness. I am just quoting an example. Alleles are a pair of contrasting characters for a particular factor, length of an individual. Genes and contrasting genes. Okay. So the allelic frequency anta use ಮಾಡ್ತೀವಿ. Not just gene frequency, we are now using the term allelic frequency. So allele for length of an individual, a tall, dwarf, is the 2 possibilities. Okay. So the frequency of occurrence of alleles of a gene, it remains fixed within a population. Like Indian population, they have a gene pool. Beyond that gene pool, there cannot be any other new type of genes. So it remains constant within a population. And that constant, we call it as the gene frequency remains constant, and there is an equilibrium. That equilibrium, we call it as genetic equilibrium. And what is it denoted by? It is denoted by 1. This constant value, the genetic equilibrium, is denoted by 1. See, gene pool is total genes and their alleles in a given population. We call it as gene pool. What is genetic equilibrium? So gene pool for a population is constant and there is an equilibrium, and this equilibrium we call it as genetic equilibrium for that population. It might vary with another population. So that sum total of all allelic frequencies occurring within a population, like Indian population, the sum total of all allelic frequencies occurring within this population is 1. It is denoted by 1. In your quadratic equation, you have that value as 0, ax plus ax square plus bx plus c equals 0. That is completely different. Binomial equations, binary equations, we are going to convert them into. Now, you have understood up to here, the sum total of all the allelic frequencies occurring within a population is equal to 1. That is nothing but an equilibrium exists. 1 indicates the existence of an equilibrium, genetic equilibrium. This is what Hardy-Weinberg speaks about. Now, they take an example now. P and Q, they are gene frequencies or allelic frequencies of a particular allele A. Capital A is a dominant factor and allele small a, recessive factor. So individual frequencies of allele A and allele A small a is represented by P and Q respectively. P represents for dominant allele A and Q represents for recessive allele A. That is small a. Okay. P square is the frequency of homozygous individuals. You know, in genetics you have studied about homozygous or pure tall, heterozygous tall, homozygous recessive character like homozygous BLAF, pure BLAF, and both the genes. See, we are all having two sets of chromosomes. We are diploid organisms, isn't it? We have one set of chromosomes from mother side, maternal chromosome, one set of chromosomes from father side, paternal chromosomes. We are diploid organisms. Each and every cell of ours has this diploid set of chromosomes. And in this diploid set of chromosomes, if one chromosome on the locus, what is the locus? The position of a gene on the chromosome, we call it as loci. Okay, the plural form is locus. So that it might have capital T, another might have small t. Which is going to get depicted? What character is shown in that individual? Capital T, small t, what character is exhibited in the individual? It is hybrid tall alpha. The dominant character is expressed. We have studied all this in genetics. The same concepts of genetics are here. P square, it is homozygous alleles we are having, dominant alleles, P square allele. Frequency of double AA genes occurring in both the chromosomes of diploid individuals in a population. We are all diploid individuals. We have two sets of chromosomes. Okay. Then Q square is frequency of homozygous recessive gene or recessive allele in a given population. Okay. 2PQ, what does it represent? It represents the frequency of hybrid or heterozygous capital A and small a. It can be example heterozygous tau or hybrid tau, we call it as. So 2PQ represents that. So now, according to this, what is the statement I gave according to Hardy-Weinberg's equilibrium? The sum total of all the allelic frequencies is constant and it is equal to 1. Now the sum total of all allelic frequencies, what are they? P square plus Q square plus 2PQ equals 1. So you can write it as equation 1. Replacing the values in equation 1, you get equation 2. P square plus 2PQ plus Q square equals 1. You can also write it as P plus Q the whole square equals 1. So this is what is the binary equation that we have derived for Hardy-Weinberg equilibrium. So any change from this value of 1, let us just assume that you have totaled up some total of frequencies in a population and you have got 2 or 3 as the value. There is a change. The difference between the Hardy-Weinberg equilibrium and that of the present change, that is going to show the extent of evolution that has taken place. That is what I have said. The frequency differs from expected value. What is the expected value? 1. The frequency differs from expected value. That is 1. Maybe if it goes to 2 or 3. So the difference of this frequencies indicates the extent of evolution that it has taken place. Higher the value, more the extent of evolution having taken place within that population. So the disturbance in HWE and the Hardy-Weinberg equilibrium is called as evolution. The disturbance in Hardy-Weinberg equilibrium is supposed to lead to evolution, formation of new species. Okay, so this is what is about Hardy-Weinberg equilibrium. So Hardy-Weinberg equilibrium in a given population, Hardy-Weinberg equilibrium is constant. That means this population is having the allelic frequencies, the sum total of all allelic frequencies is 1, which indicates there is stability in this population. Whenever there is a disruption in this equilibrium values, it leads to evolution. It leads to evolution. What is evolution? We are talking about formation of a new species. The change in the gene pool of the population. So what are the factors which are going to affect or change the Hardy-Weinberg equilibrium? Factors affecting the Hardy-Weinberg equilibrium, they are listed it as 5 factors. 5 factors are affecting Hardy-Weinberg equilibrium. First one is gene migration or gene flow. Okay. So what is going to happen here is, as the name indicates, migration can be of 2 types. Immigration or emigration. Okay. So immigration, they move away from the population. Emigration, they move into the population. If you take Indian population, here one of your friend lives here and he goes to United States or any other place and gets married to a local population or a local individual of that country, of that ethnicity. Okay. So there is a loss of gene pool of that individual in this population or not. The original population has lost the gene pool from that individual. He might have had certain unique gene alleles that is lost. There, in the another population where the person has immigrated, since he marries the person from that population, so that population receives new gene alleles, new genetic alleles. I am giving human examples in a very polished manner. Okay? So that is what you have to remember about. Now, from another country, an individual comes and gets settled in India. They have a different immigrants, what you call them as immigration. From another population, they have come to our original population. They get married to a local Indian person, okay, and now the gene pool from that person enters into the Indian population. So there have been many people who have invaded India and their gene pool has entered into the Indian population throughout the historical ages, from Aryan age to present age. Lot of such changes has happened in Indian Hardy-Weinberg genetic equilibrium has changed because of this population that have been happening along. It happens in many countries. It happens in many other species also. I am taking the Homo sapiens as an example and describing it. So remember, genetic gene migration, there is a change in original population and new populations allele frequency. Sometimes the changes may be so much drastic, it leads to the formation of a new species. Sometimes the change in the allelic frequency is so huge that it leads to the formation of a new species or speciation. Such an individual whose allele has changed and given rise to a new species, we call him as founder, and that effect, what do you call it as? Founder's effect. Okay? It doesn't happen in all the cases, only in a few cases it happens where a new individual entering into a population, their allele frequencies are so much different from the existing population that it gives rise to a formation of a new species. So we call that organism which has caused this as founder, and the effect in on the population which leads to the formation of a new species, we call it as founder's effect. For 1 mark question they ask what is founder's effect. You have to remember about that. So the 5 factors which are affecting the Hardy-Weinberg equilibrium are: first one is gene migration or gene flow, where there is a change in the allelic frequency of the original population as well as the new population where they go. So that allelic frequency, there is a change. Okay. So this is going to disturb. See, this changes is going to disturb the that population's Hardy-Weinberg equilibrium, which is equal to 1. That gets disrupted. Whenever there is a change in Hardy-Weinberg equilibriums, what does it lead to? Formation of new species. There is evolution taking place. Okay. Many such accumulated characters might lead to the formation of new species. Sometimes the alleles from one individual itself is sufficient to form a new species. So we call that as founder, that individual as a founder, and effect as founder's effect. Okay. Now genetic drift is by chance. Okay, it is by chance, it is not by choice. The genetic drift is gene migration occurring by chance. So the gene migration occurring by chance, we call it as genetic drift. Okay. It might be because of various factors. It might be because of continental drift. You know that marsupials exist only in Australian continent, not in other continents. Okay. Because they would not have survived in other continents because of the predators that exist there. and they could not have survived. Since it is completely separate from other continents, there is a unique flora and fauna that you find in Australia, down under. Okay, so remember about that. Then mutation. Mutation also disturbs the Hardy-Weinberg equilibrium, and that leads to formation of new species. What is mutation? Any sudden change in the structure of gene sequence or chromosomal structure, we call it as mutation. Chromosomal mutations are good, gene mutations are good. I have already discussed that the mutation theory was put forth by Hugo D. Rice, and they had worked on the plant Enotera lamarckiana. Within a short span of time, because of mutations, they were able to come across 20 new species of plant which arose from this Enotera lamarckiana plant. So I discussed it in the previous class, the mutation theory or Hugo D. Rice. See, mutation, what is there is, in a given population, there are mutations happening. So there are pre-existing, which is already existing in that population, advantageous, beneficial things, not the disadvantageous. Pre-existing advantageous mutations that are existing in the population, they go to the next generation. And if it is favorable for that particular environmental conditions, they are going to be, this sort of individuals are going to multiply in larger numbers. They have more progenies. What are progenies? Offsprings. And more such population with such sort of genotype with mutated genes which are able to survive, it leads to the formation of a new species, and that we call it as speciation. Origin of new species is also called as speciation. You should remember about this aspect of it. What is phenotype? The externally exhibited characters, we call it as phenotype. What is genotype? Genetic makeup, we call it as genotype. See, the phenotypes of capital T, and the phenotype of capital T, small d is same. What is that? Tall. Phenotype are the external characters which are visible. Genotype are the genetic makeup. It can be hybrid tall or it can be pure tall. When you are talking about that, you are talking about genotype. Genetics, madam already, she might have discussed about all these concepts, alleles and Mendelian principles. All those you have studied. So mutations, pre-existing advantageous mutations found in a population, okay, which if it is favorable, they increase in number. Individuals having such mutated genes, they increase in number. And it will be completely different from the original population that existed. So it leads to the formation of a new species, and which we call it as speciation. Formation of new species, we call it as speciation. So mutation also disturbs the Hardy-Weinberg equilibrium, and it leads to evolution. If there is no disturbance in Hardy-Weinberg equilibrium, there will be no evolution. That population will have the same set of gene pool. There won't be any change at all. Then natural selection. Nature selects the characters which are required, and they are passed on to the next generation. Okay. So reproductive fitness is more important than physical or the intellectual fitness I told you about. Reproductive fitness, wherein it carries that favorable characters to the next generation, and nature selects that. So natural selection also disturbs this Hardy-Weinberg equilibrium. See, there are certain characters or traits that we have which can be inherited to next generation. So that we call it as heritable variations. Apart from the property inheritance, the genetic inheritance also we take from our parents. So they are called as inheritance or heritable variations. Variations are the differences that exist, we call it as variation. So variations which can be inherited to the next generation, we are talking about. Heritable variations which enables better survival of an organism. They are going to be in larger numbers, they undergo reproduction, and they are going to have larger population of that heritable variation serotype. I discussed about the industrial mechanism in peppered corn. See, after industrial revolution, the dark colored moths, they were selected by nature because the trees were covered by carbon soot, there were no lichens. So only the dark colored moth were able to survive from the predators. So that is the heritable variation. Dark colored moth had the heritable variation which enables them better survival. And you found at that part of time more number of dark colored moths. So they had better survival, they underwent reproduction, and their population of dark colored moth was more. But prior to Industrial Revolution, the light colored moths were more in number because lichens were there and they could camouflage themselves. And at that part of time, the dark colored moths were very less in numbers. Okay. So what is going to survive? Which type of variation, heritable variation can survive is decided by nature. So natural selection also disrupts Hardy-Weinberg equilibrium. This is also one more factor which affects the Hardy-Weinberg equilibrium. So this is what you have to remember for your study of genetic recombination. Can you tell me when genetic recombination can take place? Meiosis ಆದಾಗ, you know that during meiosis there is crossing over of paternal and maternal chromosomes. The point at which they cross over, what do you call that point as? Chiasma formation. Okay, chiasma. Non-sister chromosomes, they are chromatids rather, they are going to cross over. As a result of it, a fragment of papa chromosome gets attached to mama chromosome to papa chromosome's fragment, it gets attached. That is, fragment of paternal chromosomes get attached to maternal chromosome and a fragment of maternal chromosome gets attached to paternal chromosomes. And this occurs, crossing over occurs only in homologous chromosomes. As a result, so there is a recombination of genes, isn't it? This happens during meiosis. Recombination of genes are occurred and they would be passed on to the gametes that are formed. Meiosis results in the formation of 4 haploid cells. Most of the time meiosis occurs in germinal cells or reproductive cells. So genetic recombination takes place during the meiosis process, especially the crossing over of non-sister chromatids and the fragmentation of them, which leads in genes being recombined. Genetic recombination also changes the population's gene pool. Okay. As a result of it, it disturbs the Hardy-Weinberg equilibrium and leads to evolution, and in the process of it, there is a formation of new species. Now, as Homo sapiens, how have we evolved from the Ramopithecus, Bryopithecus, the Homo erectus, to the present Neanderthal, to Cro-Magnon man? How have we evolved? Because of the disturbance of Hardy-Weinberg equilibrium. It has led to the formation of the species. At present, the modern man. It might not be the same. After another thousands of years, it might change whenever there is a disturbance in this Hardy-Weinberg equilibrium. Some changes might be immediately, you can find the formation of new species. Founder's effect I discussed above. Some of the changes are, it is like little drops of water make a mighty ocean in the way. Accumulated changes over a period of time leads to the formation of a new species. So there can be a gradual process of formation of new species, or there can be a sudden formation of new species. Both might happen. So this is what you have to remember regarding the very important concept of Hardy-Weinberg equilibrium.